Yeah, so let's now talk about the experimental groups, but we talked about it. I gave you a hint already. So these are the members of the experiment that will be receiving some sort of special treatment during the experiment. Hmm? For instance, you feel like sleeping leads to passing. You remember the Wentz hypothesis when we were studying coordination, where you say that auxins lead to stem elongation, or lead to growth of shoot, whatever. You remember, that's hypothesis. So you have to conduct an experiment to prove to us that really auxins are responsible. Auxins are growth substances. So you have to provide groups, okay? And experimental groups, those are the ones that will be given special treatment. Those are the ones that will be given enough auxins. And then you also create another group that is not given any auxins. And that will be a control group. So we are going to see what all means. For example, in the same experiment, or in our experiment, you're going to grow corn and valvet leaf plants under controlled conditions for roughly of a period of 44 or 45 days. And this is the corn, the common corn. You know, this is a C4. Let me give you the theory. Because when you carry out an experiment, you need to keep the theory within you. And then this is the valvet leaf plants. Okay? You can see how. So this is a C3 plant. Okay? And so these two plants, they will behave differently to carbon dioxide concentration. So you keep these groups. Okay? For instance, you make groups of, actually in this experiment you can grow them separately, and then after you grow them together, and you keep varying the carbon dioxide concentration. Okay? You keep varying the carbon dioxide concentration. So that's what will make an experimental group. And the independently manipulated variable, now it is that factor. Actually, usually there are factors which affect the life processes. It is that factor that can be changed purposely to investigate its effect on the life process, on the dependent variable. And in our experiment, for instance, these plants, you're going to divide them into 3 groups. Maybe you're going to grow, first of all, they are grown the same or in the same locality, same habitat, okay? And you divide them into 3 groups. You have valvet leaf plants, and you have the corn in this group. The same thing, you have the valvet leaf, and you have the corn in the other group. And the same thing, you have the valvet leaf in this group, and you have the corn. So that's how you've designed your experiment. And then you expose each group to different carbon dioxide concentration. Maybe you can give this one 300 carbon dioxide concentration measured in ppm or parts per million. And maybe you give this one 600 ppm. And then you give this one maybe 900 ppm. Okay? And then you study the effect of carbon dioxide concentration on their general or their overall productivity. That's all, guys. So the carbon dioxide concentration becomes the independent variable or the manipulated variable. So different concentration of carbon dioxide in the air, for example, 350 ppm, 600 ppm, and 1000 ppm. They are supplied to these three groups, whereby group A is given 350 ppm, group B given 600 ppm, and group C is given 1000 ppm or parts per million. That's how carbon dioxide concentration, or that's how the concentration of these substances that are so small, that are minimal, are measured. They are measured in parts per million. Then the dependent variable, now it is that factor that will be measured, or it is that measurable factor in an experiment whose effect or whose response is dependent on the change of the independent variable. For example, when you measure different, or when you provide these different carbon dioxide concentration, the productivities will be different. And in this case, the productivity is going to be measured as the dry mass of the corn and the valvet plants grown at 3 different carbon dioxide concentration. And with a dry mass, we mean the averages, the average mass for both leaves, stem, and roots. Okay? They were groups of 888 plants. 8 corn, 8 valvet leaf. I got this experiment from Campbell. Okay? It was testing the effect of carbon dioxide concentration. So I really picked interest in it. Okay? I really picked interest in it because it really answers our question. So the values, they are measured as averages. Okay? And after getting the values, you present them in a table, as we are going to see. Now you have the control group, because now see, if you say that, for instance, sleeping leads to passing of exam, okay, and you provide a given group with maybe enough sleep, okay, and then the other group, maybe you have just one group, you have just one group, and you provide the one group with enough sleep, and then that group passes. Now you cannot convince us. Maybe we shall say that maybe the exam was very easy, or if the other group fails, assuming the other group, sorry, that same group that you subjected to enough sleep fails, we shall not deny you, we shall not reject your hypothesis. Maybe the exam was so hard, okay, that's why they failed. So in order to convince us really, you have to divide into different groups, and of all those groups you must have the control group. Okay, and the control group is that group that won't be receiving any special kind of treatment. Okay, and maybe those are the guys that you leave to suffer normally, no sleeping, they go and do it there on their own. Those who are to read, they read. Those who are to They do it on their own. That's the control group. And then after you give them the same paper. Now if these guys who slept, if they have an average value maybe of 90, and these guys who read and did their own things, their average percentage maybe is 30, then we shall be convinced. So you really need a control group. So also here, the control group is among those three groups, or among the groups that you have formed. There will be a group of plants that will be subjected to normal carbon dioxide concentration. And the normal carbon dioxide concentration is 400 ppm. Actually, it varies. It ranges from 400 to 600 parts per million, depending on the environment, depending on the nature. Maybe the altitude, maybe the levels of pollution, and stuff like that. Okay? It will vary. But the normal, it ranges from 400 to 600. So that group that will be subjected to that normal carbon dioxide concentration is gonna be a control group, where other changes will be referred to. And so after having a control group, then you need to have other factors that must be kept constant, so that only that factor in question is the one that is tested. Okay? Because imagine you are studying the effect of carbon dioxide, and then light intensity changes. Some are put in shade, others are put in light, others. Obviously, you won't get the results. So you should make sure that as you are carrying out such an experiment, these other factors must be kept constant, however, in optimum ranges. And these other factors include, number one, you can have type of plants. They should be the same for all the three groups. Type of soil. Yes, all of them must be put in the same type of soil, so that don't be like, man, this one, no. Amount of light that each plant is receiving, the containers, yeah, maybe some are put in metallic containers, others are put in. So somebody can complain about it. The amount of water, should make sure that when you are to add 1 jerrycan per day, it should be 1 jerrycan for all. Okay? Yeah, uniform light is the same thing. Yes. And then finally, you have to make observations. Okay? You have to make observations. And those are the responses given by your groups, by your experimental groups, due to changes in the independent variable. And so observations can be in form of, it could be qualitative data, and with qualitative I mean it's not measurable. Not measurable. Okay? But you look at the quality. How? We have qualitative data. That one, it cannot be measured. But it involves feelings. For example, if it's beauty, maybe, how are you going to measure beauty? If it's ugly, how are you going to measure ugly? Which other factor? Think of them. So we have qualitative research and quantitative research. But usually we find all ways, okay, to convert data into quantitative data. That one is the one which can easily be measured. But qualitative, it is subjective. It has too much feelings. Okay? It is subjective, it is deep. Sometimes somebody might neglect the science and focuses on the feelings. And then data is collected when still you can have data that is quantitative. You have numbers, you have figures. Okay? And basing on the figures, for example, such our experiment, we can have such figures. And these are the average dry masses. And so basing on these figures, you can analyze them, plot graphs, do what, and you come up with a given trend that you can use to make conclusions, as we are going to see.